I'm Dr. Lauren Saplicki-D'Antonio. I'm an environmental engineer focusing on using molecular biology techniques to solve environmental problems. In this video, I'll show how we can use a DNA-based approach in the solution to solve the problem of detecting ROD in asymptomatic trees. So, my solution uses techniques called direct multiplex PCR and gel electrophoresis to display presence-absence through visual patterns. This method has been around for about 30 years, so different components have been built out and minified to apply to different scales and applications.
Now, PCR is a common acronym for polymerase chain reaction. This technique amplifies specific gene regions until there are enough copies to be seen without the aid of a microscope. Gel electrophoresis uses the pores in a gel and the negative charge of DNA to separate DNA based on size. If there are many copies of a specific gene, it'll produce a band when they all clump together at the end of the separation.
Since researchers have already developed quantitative PCR techniques for both of these ROD fungi, I assume that the genetic sequences are known and they can be used to design qualitative, more presence-absence tests for regular PCR. Multiplexing here means targeting two genes with one reaction. In the solution I propose, the two genes will indicate each ROD fungus, and one gene will be longer than the other, so they can be distinguished by size after gel electrophoresis. From start to finish, my solution yields results in a few hours. So this solution can be developed in the course of a few months with a few experiments. One, to design components that target the unique gene regions of each ROD fungus, and this is a routine task that can be done inexpensively by any number of commercial companies. The other experiment that's needed is to verify that the direct part of the solution works. This would involve macerating a tiny bit from either a leaf or a flower petal or a brassy stem from a tree that has ROD of each type and putting it directly into the reaction at the start. If the direct part does not work, there's a simple and inexpensive 30-second protocol that can extract DNA and be inserted into the solution before the PCR step. So, to recap, this solution relies on established techniques that have been miniaturized for field use and can be performed using battery power with either the mini PCR system or the bento lab. Components of this solution have been validated in the field, and those that have not are routine tasks with established protocols.